Oh, it's annoying.
All right. So today we'll start doing applications and strategies, techniques, interventions, whatever you want to call it, any number of synonyms. How to, all those pre and post, like this is pre, P90X, or this is pre whatever supplement, or pre something. And they always have their post picture. To be able to facilitate healthful rapid weight loss, there are some tricks. If you do too fast of weight loss, no matter what tricks you're using or how carefully you strategize, you're going to find metabolic deficits. And so pacing actually is one of the things we'll talk about. I didn't update the points yet, but we have a picture. We have a picture of the points. So I haven't updated that page yet. And remember on Friday, get the team names. Whatever your team name is going to be, the most creative one gets points. I will put up, just clinicalphys.com, I'm going to put up there, after every single lecture, once there are PDFs, you don't need to read the PDFs. It's not like I'm holding you accountable for things that the PDFs say, but if you decide you want to know more about a topic I'm discussing, if you want to know more about special populations, whether it's diabetes, it's heart disease, whatever, if you want to know more about kids, I put all of those PDFs that are most critical or most used in my lectures. I just make them available. And so there's the fat metabolism ones, and there's going to be about 20 PDFs in this slideshow that will be uploaded. Again, you don't need to read them, but if you're interested, if you want to go read more about the supplements that I will very briefly go over, if you want to know more about thermal regulation and body composition, things like that, all of those PDFs are in there. But I'm also going to put that checklist of stuff to know, all of those there soon. So you can pull up the PDF and just see the checklist of stuff to know slides. So you don't need to fast forward through the videos to see them. So I'll get that up pretty soon. But just remember, we have different body types. Do you guys remember the whatever morphs?
Just
that one, the machinist, yeah. Christian Bale, like pre-Batman.
Oh, you want?
I thought I heard your voice.
Oh,
So an endomorph, mesomorph with an E, an ectomorph. So these three different. Celeste, what's your team? Team 1, and then I'm just going to go 2 through 5, 0.01 and 0.02. Because I don't know who was talking, but these different body types, a lot of this is genetic, but we can play within our genetic assignment. We can make changes within our genetic assignment. So if you are a mesomorph, you might have in this healthy obese category, owing to an increased amount of lean body mass. And right here, just something like normal BMI, right? Princess Diana, or normal BMI, but that's going to be skinny fat. There's a presentation of adiposity that is not associated with health and ongoing, warding off disease and entering into those later years as healthful as one would like. So what's metabolism? Summation of all chemical reactions. Some of them are catabolic, some of them are anabolic. What do we know? Amanda, what's your team? 2? You said that with a question mark. It's 2.009. So when we talk about metabolism, is this just like calories in, calories out? No. Give me some examples. Some examples that would overturn that premise of calories in, calories out. Thyroid hormone. Yeah, if someone is hyper or hypothyroidism, if they have that, you're going to see a bunch of sweaty, sort of agitated movements, some NEAT activity, non-exercise activity, thermogenesis, and the high body heat for hyperthyroidism. And hypo, there's not enough snuggies in the world to keep warm. And so that's the basal metabolism. What else? Menopause, yeah. Aging and menopause. Menopause is a combination of aging and changes in hormones, but are these sort of hormone effects, whether it's testosterone, it's estrogen, it's growth hormone, whatever these hormones are doing is going to affect your metabolism regardless of how much exercise you're doing, regardless of how mindful you are with your NEAT, regardless of what you're doing with your diet. Now, those things will affect your hormones, but your hormones kind of make up their own minds to a degree.
Should we know that graph with percentages of like TDEE is BMR and thermic effect of food and the percentages? Should we know that?
Not that exact percentage. I have it in these slides.
Mm-hmm.
You don't need to know like 60% is basal metabolism because it depends. It depends on the person, depends on the population, it depends on the conditions, the circumstances of the population it's in. So that's going to depend. The ballpark figures that the thermic effect of food and exercise is pretty similar. Non-exercise activity thermogenesis is bigger than that. And when we start looking at the big explanation for our waist circumference and our BMI, the body composition, however you want to break it down, metabolism, in particular basal metabolism, of just what your body is doing on its own without your input, without your manipulations. That's the big ticket item. All right, what's that? ATP. I couldn't see whose mouth. Like no mouths are visible. What's the reaction here? So yeah, if we're going to do ATP hydrolysis, yeah. So that's ATP hydrolysis, hydro, as in H2O, lysis, as in we're going to lyse an ATP. We're going to lyse that ATP, right? So what goes in? ATP and water, and what comes out? ADP and phosphate and hydrogen ion. Remember, because we take the H2O, there's an O and an H is going to plug up this, and then the other H goes floating off. And at the end of the semester, we'll talk more about that H. So ATP hydrolysis. We are lysing our ATP using water to do so, and that's our energy. That's where we're getting it from. What's this one? Yeah, the creatine kinase. Yeah, so back here is ATPase. So ATPase, and one of the first times I taught 129 was he was a water polo player, and I think he was not engaged in class, and it came to the test. ET paste. Okay, like Elmer's version of metabolism. Yeah. Sure, but ATP ACE, all one word, ATP ACE. And then over here, the creatine kinase reaction. So creatine kinase is the enzyme, and what goes in? Perfect. Yeah, so a hydrogen ion, that ADP, because we're going to phosphorylate it, we're going to attach phosphate to the ADP, and then the creatine phosphate or phosphocreatine, however you want to say it. And the supplements do work, but we're not going to talk about that today because we're not talking about performance supplements today. We're talking about weight loss supplements, fat-burning supplements. And what comes out? Creatine and ATP. Wait, one? Did you want to donate to Team 2? 022. Yeah, the creatine kinase reaction. So creatine kinase is the enzyme, and it's going to just creatine donates, phosphocreatine or creatine phosphate donates that phosphate to the ADP. So you get your ATP back and you consume a hydrogen ion in the process. And then you just wind up with ATP and that creatine. All right, what's this? What are we looking at?
AMPK can't cure many things.
So it's a little bit complicated the way it shows because we're showing the actual structures, but we have ADP, adenylyl kinase, and then we're going to get our ATP and AMP. Yeah, so it's Robin Hood deciding who to make rich and who to make poor. It's sort of an inverted Robin Hood. Everybody's middle class, we're going to make some people rich and some people poor. That's adenylyl kinase.
Wait, what was the enzyme for creatine?
That creatine kinase.
Oh.
Yeah. So the creatine kinase reaction is creatine kinase is the enzyme. So ATPase was the first enzyme. Creatine kinase is the second one. Adenylyl kinase, not adenylyl cyclase. What does adenylyl cyclase do? Cyclic AMP stuff. But adenylyl kinase, that reaction. Echo 19. Okay, what's this? No. There. Yeah, just glycolysis. And we're going to talk a lot about glycolysis, and one day, at some point during the semester, the only really important thing to know is this is beginning with glucose here. It doesn't have to begin with glucose. And a human being who's exercising, we're sort of ignoring this step, and we're using glycogen instead. But also, at the end of glycolysis, what's the end product? Pyruvate. What do you do with pyruvate? Yeah, you can, it depends. You can turn it to acetyl-CoA and throw it in the Krebs cycle, or you can convert it into lactate. We'll talk all about lactate later. But let's pretend you convert it into acetyl-CoA. How else can you get acetyl-CoA? Beta oxidation, right? Let's start with lipolysis before we get to beta oxidation. What's lipolysis? How does that work? Good. Yeah, so we have a glycerol and then 3 strings of fatty acids. They're probably 16 or 18 carbons long. They don't have to be, but the bulk of all of your fatty acids are 16 or 18 carbons. How does that stimulation, what gets that going?
Beta adrenergic receptor, epinephrine, or norepi.
Okay, good. So we have epi or norepi. Epi, we think it's a hormone, right? It's a hormone in the blood. It's the mean hormone. It's an endocrine system. Norepi, this is coming out of the nerves. Fat is innervated. Nerves, contact fat. And so both epi and norepi can mobilize your free fatty acids. They can do lipolysis. They can lyse the lipids, lipid lysis. So epi, norepi, bind to a beta adrenergic receptor. What's the next enzyme in this?
Oh, enzyme.
Adenylyl cyclase. Adenylyl cyclase, and that does what? Yeah, I knew what you meant. So we convert ATP into cyclic AMP. Cyclic AMP binds to pKa. Then what does pKa do? I don't know who's talking. Yeah, so hormone-sensitive lipase really gets that one going. It gets adipose triglyceride lipase a little bit, but really gets hormone-sensitive lipase going. And perilipin. And what's perilipin? Perfect. It's a coat. Like you unbutton the thing and open it up and whatever. Now you have access to the lipids and to the triglycerides, a triacyl glycerol. And so the first one, what's the enzyme that really does the first one? ATGL, adipose triglyceride lipase. And so now we have a diacyl glycerol. What's the hormone that does that one? Hormone sensitive lipase. Now, again, there's other stuff. There are other enzymes. There are other fates. Diacylglycerol kinase we'll talk about soon in the mTOR reaction where we can convert this into phosphatidic acid. But hormone-sensitive lipase, we now have another fatty acid floating off. What's the last one? Mono. Yeah, monoacylglycerol kinase. And so let's get rid of that last one. Now we have fatty acids that are floating off, right? They're headed toward, you release them from the cell, and they're headed somewhere, right? They're headed towards some metabolically active tissue, ready to be beta oxidized.
So the adipose triglyceride lipase, ATPL, what's that again?
It's the enzyme that does that first step.
And you said PKA is mainly activating hormone-sensitive lipase?
Mainly activating hormone-sensitive lipase. There's pathways with like AMPK, and so there's, when people talk about hormone-sensitive lipase, it's a regulatory step. Leptins are regulatory, so everything's sort of a regulatory step.
So where does that adipose triglyceride lipase, how's that get activated?
AMPK is one way. There's several pathways for activation. PKA actually does activate it, not very pronounced.
Okay.
It's like a 13th as much as it activates hormone-sensitive lipase.
You kind of need that to get to the second step, right?
You need it, yes, but you can have it though. It will be doing its duty, but it's not quite through the same sort of canonical signaling of PKA.
Okay.
So lipolysis is done. Insulin again totally blocks it. Insulin totally blocks it. How does insulin block that?
Adenyl kinase cyclase.
Yeah, some words in there, and some of it's on the right track. Yeah, phosphodiesterase, PDE, and then converts to cyclic AMP into regular AMP, and AMP doesn't activate PKA. So that's one of insulin's obstructions, one of its inhibitions of lipolysis. Another one is genetically, insulin will stop or at least inhibit the expression of adipose triglyceride lipase, so you have less of that enzyme. Okay, so lipolysis, we are sending fatty acids through the bloodstream. Fatty acids are going through the bloodstream. Then what?
Doesn't that get that's good oxidation converts it to acyl-CoA, right?
Yeah, so a little transporter, we're gonna get it into the cell and we're gonna attach coenzyme A to it. So we have acyl-CoA. Now, an acyl-CoA cannot just get into the mitochondria. It can't just run on in there on its own. An acyl-CoA can. What do you have to bind to it? Carnitine. So you attach carnitine to it. And once you do that, you can shuttle it in. But you don't have acyl carnitine, which is what comes in. So acyl carnitine. And then a regular carnitine goes out at the same time.
Wait, can you go from the fatty acid to how did you get that from the fatty acid?
There's a transporter protein that shuttles it in, and at the same time, you are attaching a coenzyme to it at that same step. So you go from just like your fatty acid, attach a coenzyme A, you shuttle it in, and then attach coenzyme A to it. And now you have an acyl-CoA. Acyl-CoA is what but you're in the cell, but you're not in a mitochondrion yet. In a mitochondrion is where beta oxidation happens. So attach carnitine to it, acyl carnitine. You can send that acyl carnitine into the mitochondrion on a transporter. And then free carnitine goes out. You take that carnitine off, and then you run it through beta oxidation. 4 steps in beta oxidation. Anybody remember what those are? 2 of them are the same thing. Dehydrogenation, hydration, and dehydrogenation again, and cleavage using thiol. Just like ATP hydrolysis uses water, and this uses thiol. But a cleavage. So you do for beta oxidation, once you have your acyl carnitine back, or your acyl-CoA back, you've removed the carnitine. The free carnitine comes back out to shuttle more acyl-CoAs in. You have your acyl-CoA back. You do a dehydrogenation step, hydration, another dehydrogenation, and then you cleave off an acetyl-CoA. So now we've mobilized our fat. We've taken, let's say, the 16 carbon lipid, and a 16 carbon fatty acid. And how many acetyl-CoAs do we get out of that? But out of it, so if we have 16 carbons.
Oh,
1, 2, 3, 4, 5, 6, 7, 8. What happens if we have one more carbon? What happens if we have the unlikely? Yeah, we get one fewer. Well, there's one because the last one is not an acetyl-CoA, right? That enters the Krebs cycle as an intermediate. Okay, so now we've made a bunch of acetyl-CoA. What do we do with that? It goes into the Krebs cycle. Yeah. What does it bind to? Binds to oxaloacetate. Was that Alex's mouth? I didn't know where you were going. So it binds to oxaloacetate. So here's acetyl-CoA. Binds to oxaloacetate, makes citrate. That undergoes a bunch of changes, a bunch of enzymatic changes. 2 of those, you release carbons as carbon dioxide, but you also make that stuff. What does that stuff do?
Enters into the electron transport chain that carries electrons.
Perfect. Yeah, these electron donating compounds that are going to donate into the electron transport chain. So that's TCA cycle, tricarboxylic acid, Krebs cycle, citric acid cycle. It's all the same thing, different words for the same thing, et cetera. Right? That's the electron transport chain. And we could say, oxidative phosphorylation too, because we need ATP synthase at the end. Oxphos. Where does NADH go in? Complex 1. Where does FADH go in? Complex 2, right? And at complexes 1, 3, and 4, you're pumping protons, hydrogen ions into that intermembrane space, and that's what's going to power ATP synthase. And we get a ton of ATP that way. So that's a complete use of fat from the time that it's living in a tummy, that it has that midriff real estate, until the time that it's just completely gone and we have water and ATP. That's the whole course. So again, lipolysis first. What that mean? Says Ryan Gosling. What that mean? What's lipolysis? Lysing that thing, right? And then remember, insulin binds to its receptor. You go through PI3K, PKB signaling. There's PDE. PDE is converting cyclic AMP into regular AMP. Regular AMP does not activate PKA. So if you have a beta adrenergic receptor, and there's your adenylyl cyclase, epi or norepi binds to that, deactivate that thing, convert ATP into cyclic AMP, that binds to PKA. PKA phosphorylates, activating tons of stuff, including pyrrolipid and hormone sensitive lipase, a couple of those regulatory steps in lipolysis to move stuff along. And then getting that fatty acid, once you have gone through lipolysis, remember, getting that fatty acid into a mitochondrion, get it in, convert to acyl-CoA into the cell. And then convert that acyl-CoA into acyl-carnitine, attach carnitine to it. Move that acyl-carnitine from the cytosol into the mitochondria. So once it's connected to its usher, you shepherd it in to a mitochondrion, which is where beta oxidation happens. It can't get there on its own. Remove the carnitine from the acyl-CoA, and now that acyl-CoA, that fatty acid that you mobilize, that now has coenzyme A on it, is ready to undergo beta oxidation. Beta oxidation is, this is 14, right? So you go to 12, you release an acetyl-CoA. Then you go to 10, you release an acetyl-CoA. You go down to 8, you release an acetyl-CoA. Go down to 6, and then that last one, that last reaction, as you go through the steps of beta oxidation, that last reaction is you get 2 acetyl-CoAs.
Each time it breaks off a CoA, it has to go back out of the cell and back into the cell.
You just see it like back up here. So
it stays in the mitochondria.
yeah, right back at the start. So it's like a cycle too. Everything is sort of cyclic. But it just undergoes those 4 steps. But the thing to look at here, remember, fatty acid comes in. It's transporter protein. You attach a coenzyme A to it, right? So we have acyl-CoA. Then we attach carnitine to that acyl-CoA, and so we get acyl-carnitine. Acyl-carnitine is able to enter the mitochondria. Acyl-CoA can't. Acyl-CoA can live in it, but it can't enter it. So you create acyl-carnitine, carnitine goes in, acyl-carnitine goes in, regular carnitine comes out, ready to attach to another acyl-CoA. Remember the idea of ushering somebody to their seat, and you have them sit down, and then you have to leave the venue, you have to leave the tent, the whatever it is, the church. You have to leave that little section and go get another grandma and bring that grandma in. And so wedding seating is in here, and carnitine is ushering everybody to their seats, but has to come out here and grab one, send them to their seat, come out, grab another, send them to their seat. And you have to be with carnitine to get in the door. That's like your lunch ticket, that's your passport, whatever. But then you remove the carnitine and then undergo those 4 steps in beta oxidation. So those 4 steps being dehydrogenation, dehydrogenation, hydration, dehydrogenation again, and then cleavage. And with cleavage, you get that acetyl-CoA. The acetyl-CoA can enter the Krebs cycle or ketogenesis. If you don't have oxaloacetate in the Krebs cycle in the liver, you can make ketones out of them. So these are just the 4 steps, same stuff, right? Dehydrogenation, hydration step, dehydrogenation again, and then that cleavage.
Where does the, like if you had a 17 carbon acyl-CoA, what happens to that last one that doesn't get converted?
Succinyl-CoA is what it gets converted into. I guess it wasn't on the slide. I was hoping it was on the next slide and I could just click, and that would be much smoother than clicking here and having it not be that. Succinyl-CoA, we'll get there in a sec. So this is another look at what's happening. You get to the end and you've shortened it by 2. That 2 is an acetyl-CoA, and then the other thing just keeps undergoing beta oxidation. Just keeps undergoing. It's now just a shorter one. Remember, most of your fatty acids, these are 16 or 18 carbons. Well, there's 16 carbons as soon as the 18 carbon one is finished its first cycle through beta oxidation. Okay, here for the odd carbons. And so succinyl-CoA. Oh, so propionyl-CoA is what it immediately gets converted into, and then the end product is succinyl-CoA.
So it's kind of better because it's sitting in.
There's part of it that's better. Yeah. Remember the Krebs cycle, there's a lot of ins and outs with the Krebs cycle. I'd be happy to have, give me some more of that. I'd be sure. Yeah. So, rather than splitting a 4-carbon compound into two 2-carbon compounds, you get a 2-carbon and a 3-carbon. The 3-carbon does not become acetyl-CoA. It's to become succinyl-CoA. There's the Krebs cycle, right? So if the acetyl-CoA comes in, this 4-carbon thing up here is going to be oxaloacetate. The 6-carbon thing is going to be citrate. You'll lose a couple of carbon dioxides. Those are the acetyl-CoA carbons. So you shed those 2 carbons and return to a 4-carbon compound, and with oxaloacetate again. But you get the NADH, FADH, ADH, NADH. You get those along the way. And then remember, these are donating electrons. These things donate electrons. Where are they donating? To the electron transport chain. So which one, NADH or FADH, which one is Wallace and which one's Grimes? NADH is Wallace because it comes in earlier, so it associates with even more hydrogen ions being pumped into that intermembrane space. Even more hydrogen ions are pumped. FADH, you pass that first step in the electron transport chain, and 4 hydrogen ions get pumped into that intermembrane space there. And so you don't get those 4 hydrogen ions with FADH. FADH, you don't get as much of a gradient difference. Just another look at these 4 complexes. Cytochrome C oxidase, this is the last one. This is complex 4. And so we're going to make oxygen, or we're going to bind the oxygen you're breathing. We're going to bind protons and electrons to it to make water. So that's cytochrome C oxidase. What's an inhibitor of cytochrome C oxidase? Cyanide, right? You can get it from amygdalin in your apples. And so just another look at the electron transport chain. There's complex 1, you see NADH donating its electrons, hydrogen ions being pumped. Complex 2, you see FADH donating its electrons, and no hydrogen ions being pumped. Complexes 3 and 4, you see hydrogen ions being pumped. 4 here, 4 there, 4 there. You get this big gradient in this space, and this is how they get back ATP synthase, and it powers the production of a bunch of ATP. So that's oxidative phosphorylation. The electron transport chain is really just that 1, 2, 3, 4. And number 4, cytochrome C. Now those are the same thing, right? So electron transport chain, complexes 1, 2, 3, 4. Complex 1, NADH. Complex 2, FADH. Complexes 1, 3, and 4 pump protons, ATP synthase. That's oxidative phosphorylation. Let's make a bunch of ATP based on that gradient. So cyanide, right? That's going to be cytochrome C oxidase. The pyruvate dehydrogenase complex, that's what arsenic is inhibiting, is the pyruvate dehydrogenase complex. Apple seeds, despite what Orange is the New Black says, you don't get arsenic there, you get more contaminated drinking water. This is an amygdalin conversion into cyanide to block cytochrome C oxidase. BMI, we've already talked about this a little bit today, right? Or BMR, rather, basal metabolic rate, the bulk of all of your metabolism is going to come from your basal metabolic rate. Now, if you're a marathon runner and you did a marathon today, this percentage is going to be a little bit different, right? If you have a ton of meat or no meat at all, that percentage is going to be different. That's thermic effective food. Are you at the buffet eating nothing but meat? That number is going to be different. Are you fasting? That number is going to be different. So don't write any of these numbers down. These are just ballpark figures. But exercise, you know, you're like wrestling, it's like Herodotus inspired wrestling. You're jumping rope in a very patriotic way. Whatever it is you're doing, your exercise, this is a tiny fraction. This controllable, scheduled exercise is a tiny fraction. Most of your consumption is coming from organ function, your brain in particular. The brain in particular, that's the hugest consumer of your ATP. Thermal regulation, huge. Maintaining ion gradients, huge. And it varies, right? The ability to maintain these things, they leak, and you have to pump them back depending on a metabolic state. You can be efficient or inefficient. So calories in, pretty controllable. All right, that's a pretty controllable behavior. Calories out, you can control so little of that. The more you try to exert control, the more it changes in the other domains. You exert control over like maybe 10%, and like 90% of it just starts changing. And that is really difficult to manage. So there are some good strategies. There are some ways to try to manage it. First, the types of fat. We've all heard of brown fat, right? We used to think like, well, babies have it. Well, it's in adults too. Adults have it too. But white fat, brown fat, or white adipocytes, that's fat, or brown adipocytes, and beige adipocytes. Sort of an in-between, kind of a hybrid. When you look at ultrastructural morphology, you see low mitochondrial content in the white ones. Low mitochondrial. Look at the beige, high mitochondrial content. You can convert some of the whites into beige. There are ways to convert white fat into beige. And you know, different amount, when you see BAT, brown adipose tissue, BAT, so what's WAT? White adipose tissue. So in all these things, when you see BAT and WAT, that's what that stuff stands for. But it changes with aging. This stuff changes with aging, and this is sort of what it looks like. Infant, here's a bunch of brown fat. Okay, those are some regions where it tends to be. It's not like this is every single person exactly. This is the diagram. This is exactly what an infant looks like. Infants have huge heads and stuff. Young adults, we're starting to see some visceral adipose tissue. And there's subcutaneous adipose tissue. And that changes a little bit. You know, aged, less subcutaneous than there, more visceral. But then you look at the brown fat characteristics, aged people, less brown fat. less brown fat, tend to have more white fat. Brown fat is really nice for thermal regulation and maintenance of metabolism. We don't have as much as we go through life, but we still have some, and we can convert some of that white into beige. Just a few passages to pay attention to here in this article it will be on that website. Adipose tissue is now known to express and secrete a variety of bioactive peptides known as adipokines, which act at both a local and systemic level. So fat is not just a famine fund. Fat is not just there waiting to be used in times of starvation. Fat plays endocrine roles. It's an endocrine organ. It secretes things. Fat also, in addition, has numerous receptors that allow it to respond to afferent signals from traditional hormone systems as well as central nervous system. Fat has a bunch of receptors on it. You know some of these receptors, what are they? What have we been drawing on fat? Epinephrine, yeah, beta-adrenergic and insulin. I mean, there's a lot of these receptors. But through all of these roles, right, as having all of these receptors and secreting all of these hormones, fat is involved in metabolism, the neuroendocrine function, and immune function. Fat plays roles. And so if you get too low in fat, you're going to encounter problems, especially with lady folk. That body fat percent has to be a little bit higher. Otherwise, the problems start setting in. You don't need to write any of these things down, but you should probably recognize some of them. Leptin. What's leptin?
It's a hormone in the brain that's released that makes you not hungry.
So there's leptin and ghrelin. Leptin, yeah. So we think it's released from fat cells as they're growing. So as fat cells are growing, we release leptin, and it acts. Your hypothalamus controls almost everything. Your hypothalamus controls a great many functions, and among them, appetite. And so if something is regulating appetite, it's probably acting on the hypothalamus. Both leptin and ghrelin do. Leptin is going to suppress appetite, or at least an absence of leptin will make you very hungry. Leptin as a supplement, I didn't even include it because it's just so ridiculous. In the list of supplements that don't work, it's not even, there's no point in listing leptin, even though you can buy it as a fat loss supplement. It's so pointless, it doesn't do anything. When it first came out, when we first identified it, when we first recognized it, that was like breakthrough. We have cured obesity forever and always, and everybody ever. And then it turned out to just not work because there are so many inputs for appetite, and leptin is just one of them. Ghrelin, if you have ghrelin, that's going to be released from the gastrointestinal tract, from the stomach, when it's all shrunken. You're going to release ghrelin, and then ghrelin is going to act on the hypothalamus to give you the munchies, food-seeking behavior, stuff like that. But leptin is released from fat as it's growing. And it's, I guess you've had enough. If your fat cells are growing now, you've probably had enough to eat. Leptin is critical for signaling puberty. So we see kids getting hitting puberty at younger and younger ages. Look at a gymnast who's like 35 and prepubescent. They're like ripped with veiny abs, and they look 7. If you're really lean, your fat cells have not had a chance to be expanding and releasing leptin, which is one of the triggers that kisspeptin. There's other stuff involved, but one of the triggers of puberty is leptin. So kids at younger and younger ages, as their fat cells start to expand, when you see childhood obesity, fat cells expanding, you're releasing leptin earlier and earlier in life, and we're triggering puberty earlier and earlier in life. So fat is what this major trigger for when puberty sets in. Tumor necrosis factor, TNF alpha, interleukins, there's just adiponectin. There's all this stuff in here that's critical for metabolism and general function. Just another look at BAT and WAT, visceral white adipose tissue as compared to subcutaneous white adipose tissue, and some of their general roles, like core body thermal regulation in BAT. For the subcutaneous, we have the insulation, energy storage, some adipose secretion. There's slightly different functions for each of these. What we store in excess, what abdominal adiposity and you know, like the triceps and stuff, that's a bunch of white adipose tissue. And this is really what people are trying to discard, to dispose of, is white adipose tissue. But stuff can brown it. You can take white adipose tissue and beige it. You can brown it. So for the browning, right, you're going to get more thermogenesis. It's more sensitive to insulin, less inflammation, reduced association with metabolic disorders. The more you move in this direction, the healthier your fat is. The less associated with disease conditions, pathological conditions, your fat becomes. And so people are trying to figure out exactly what it is that makes white cells beige. Beige is a verb, become beige. And one of the things, you know, prolonged cold exposure, very effective. That's what most people have been studying, prolonged cold exposure. You can get it with pharmacological interventions. You can get it with exercise. There are certain nutritional compounds that you can get it. One of the conversions, though, appears to be lactate. There's a lactate-induced browning that you'll see. And so you can see this effect with some exercise. There are some studies that have found exercise behaviors to associate with the browning of our white cells.
What was the intervention for the cold therapy?
So I'll talk about this in a few minutes. There's a number of interventions that have been looked at for the cold therapy. So when I get into some of these like tips and strategies and stuff, I'll talk about it. But in short, just don't get frostbite, right? Don't get hypothermia. You can do it in the shower. You can do it in a cold bath. You can do it in something wet. And it doesn't have to be freezing. You don't have to cover in ice or anything. But get in there, it's like, this is uncomfortable, and sit there for a minute, and then get out and don't dry off. And like shiver a little bit, do some, you know, multiplication tables, whatever passes the time, right? And then get back in, just do a few reps. And you don't have to go as long as like cardio session, right? You don't have to go on the treadmill for 45 minutes. You don't have to like do that. You don't have to be like shivering and frozen and hating life for 45 minutes. So it gets kind of an in-out repetition, long reps tends to work best for one person. So getting into the strategies, the first couple of strategies are like, let's just not be crazy. So when we're looking at what very lean means, what the body fat percentage is, we can start to encounter metabolic problems, bone density, immune system problems. We can get hormonal problems. There are all these problems that can be encountered. If one will start to get below, say 11, 12%, something like that, somewhere in that range. Now this is out of ACSM. And I will get this stuff posted online so you can see these tables. Just for guiding people, when people want to lose fat, let's pick a good number first. Pick the number you want to be, and don't be insane about it. Guys can get into the single digits without really experiencing problems, but you don't want to get too low in the single digits. Women, I would say 11, 12% is about the bottom that you would ever want to be. Otherwise, you can start to encounter some problems. So the rate of fat loss. Bob Ross would do a painting in like 26 minutes. And I remember being a kid and getting frustrated that mine sucks and I don't even have that first tree done yet. And he has all the world's trees and all of them have a squirrel. And it took him 26 minutes and I've like rewound the episode 25 times and I'm still working on my first tree. That sort of pace is much more appropriate in weight loss. You don't want to be a Bob Ross about your weight loss. And because you remember what happens with The Biggest Loser, the metabolism, the metabolic crisis that follows them like an obese shadow for the rest of their lives. They just destroy their metabolism, and good luck even maintaining at that point. You're exercising all the time and eating nothing, and there's nothing worse. So 1 to 2 pounds a week, that's a good rate, right? Fat burns slowly. 1 to 2 pounds. If you're burning 4 pounds a week, what are you losing? What is leaving your body that's not fat? What what non-fat tissue are you discarding? Right, this is about as fast as you want to go. Now, at the onset, if you're sort of heavier than you're ordinarily accustomed to, yeah, you might lose, 2.5 pounds a week, something like that. Okay, and then you get 6 weeks in, and now you're like 1 pound a week. So I'm fine. Yeah, I wouldn't try to go above that. And tons of explanations of the yo-yo dieting. Now, there's mixed evidence on that yo-yo dieting. You think like Oprah. Everyone loves Oprah. She's on the cover of the O magazine and she weighs like 600 pounds. And then the next one, she's on the cover and she's like 110. And then the next one, she's on the cover and she's like 350. I mean, she was just sort of back and forth and back and forth. There's mixed evidence on how dangerous that is. There's mixed evidence on how difficult future bouts of weight loss are. So it's hard to say, here's exactly what you're up against in that sort of yo-yo situation, but it's not good, right? That's what you don't want to do it. And we all know that you crash when you lose too much. Now, getting into exercise intensity, it was once thought and said, and championed, I mean, people would boast this, that what happens is it's, fat metabolism, fat burns slowly, it's oxidated, you can't do this very fast. The fast stuff is like creatine. Glycolysis is kind of fast, whatever. Oxidative phosphorylation is super slow. So what people said was, as you increase the intensity, here's resting metabolic rate. Here's like you're walking 1 mile an hour. Here's you're walking 2 miles an hour. Here's you're walking 3 miles an hour. As you increase the intensity, the amount of fat metabolism, of fat utilization is increasing. And it's increasing and it's increasing. And then what everyone said for the longest time is it plateaus. That happens. You're going so hard and you're at your VO2 max, let's say. You're consuming as much oxygen as you can possibly consume. And so you're increasing and increasing and increasing your fat metabolism. You're like 1 MET, meaning 1 metabolic equivalent of the task, meaning your basal metabolism. 2 METs, 3 METs, 4 METs, 5, 6, you can go up like 23 METs, and you increase the amount of fat use, and then you plateau. And then as you continue to increase intensity, what you're doing is supplementing that with carbohydrates, and then you reach a plateau there too. That's the graph people used to draw. That graph is inaccurate. What really happens, here's your fat metabolism, you're doing this. You do that. At higher intensities, you actually burn less fat. Not just the percentage. The percentage changes drastically. You start to favor carbohydrate metabolism. That percentage changes drastically. But the absolute amount of fat that you burn is less as you get more and more and more intense than it is at a lower intensity. I'll talk about the reasons in just a second. So finding the right intensity that you can maintain for a while, where you do have elevated consumption, elevated lipolysis, elevated beta oxidation, where you have elevations in those things. But there is a crossover somewhere with carbohydrates increasing with intensity, fat decreasing with intensity. But this is relative contributions, but there comes a point where you're just not consuming fat anymore. Now, it depends on the person. It depends on the person. So highly trained person compared to moderately trained person. And this is the percentage of VO2 max. So you can maintain, you're up here at 65% or so of your VO2 max. Yeah, you have a lot of fat metabolism. So this is fat oxidation. How much fat oxidation you have, how much fat burning you're doing over here. 65% of your VO2 max and then it's going down. But you can get more if you're trained. Males tend to have a little bit higher fat metabolism too. A little bit more oxidation as well. But the training state seems to matter. So it's not just everyone is the same. A 9-year-old boy and a, Olympic female athlete, whatever. These are different. The ranges, so if you have maximal rates of fat oxidation have been shown to be reached at intensity between 59 and 64% for trained individuals, and 47 to 52% of the general public. So that's not that high. That intensity, it's not lying around, but you're not really pushing it that hard, and you're maximizing your fat oxidation there. If you increase, it's not simply the addition of carbohydrate, it is the reduction of fat. And this one has a nice image of, there's local fat, there's local adipocytes, and then there's peripheral adipocytes. And so plasma fatty acids, this is peripheral. If we're at 25% of our max intensity, 25% of our VO2 max, peripheral adipocytes, plasma fatty acids, that's what we're using.
Wait, is it the percentage of that for that oxidation or
The intensity. That percentage is about intensity. So if it's a trained athlete, if somebody has a history of exercise, you're looking at a pretty good percentage of VO2 max. If somebody's just off the street and like they don't appear to be in great shape, they're probably in this range, where that's the percentage of their VO2 max. You can think of this as some sort of percentage of their maximum intensity, in which maximum fat oxidation is likely to live, somewhere around there. But again, it depends. It depends on the nature of training. It depends on diet, right? It depends on a lot of stuff. So that range is sort of pliable. But plasma fatty acids, this is the stuff in the blood that you're sending. You've gone through lipolysis in some peripheral tissue, and you're sending this. Now you go up to 65%, and plasma fatty acids are actually reducing. You're reducing the contribution of peripheral adipocytes to the metabolically active tissue. But what you see, intramuscular triglycerides, triacylglycerols, that's a triglyceride, right? That percentage increases. The local stuff. So what kind of happens in the periphery is at really high intensity, you actually do seem to go through lipolysis, but they don't get released. You don't release those into the blood. After exercise, you might have a little bit of a supplemental dump of fatty acids into the bloodstream. During exercise, though, you're not really releasing them. So think about dropping kids off at school. Do you do that at 5 miles an hour or 50? 50 miles an hour, get out of the car. No thanks, I'll sit here. I unbuckled because my school's passing by at 50 miles an hour, but I didn't get out of the car, right? That high intensity, you might go through lipolysis at a fine rate, but you're not releasing it into the blood. And so the local tissue, you might consume a little bit more fatty acids at the local tissue, at the metabolically active muscle. But you increase the intensity even more, get to 85%, and you contract both of those. You limit both of those. Both local and peripheral fat metabolism starts to decrease. This one shows a little bit more. Let's look at plasma glucose and muscle glycogen, in addition to muscle triglycerides and triacylglycerols and plasma fatty acids. So this is a different study. I'm just looking at even more. So plasma glucose, not much, right? Not much. Muscle triglycerides, not much at 25%. You have to 65% muscle glycogen, 85% muscle glycogen, tons, right? Plasma glucose might go up a little bit. You're not using much glucose to support exercise. Really what you're using is glycogen for this stuff. But either you increase the intensity, muscle triglycerides go up a little bit, and then they contract. The more intense you go, the less you use. And then the plasma fatty acids just get lower and lower and lower. Peripheral use. So if you want to whole body systemic fat consumption, lower intensities are much better. If you want to burn some fat out of the target tissue, higher intensity, okay. Yeah, you'll get a little bit more out of that local tissue, but total fat consumption is going to go down. Same article, and this is looking at, I believe it's 25% and 65. Okay, so this is 25% of your VO2 max. Plasma fatty acids, that's just like everything. 25% of your VO2 max. 65% over time. So this is 15 minutes, 30 minutes, 45 minutes, 60 minutes, so long duration stuff. Plasma fatty acids compared to muscle glycogen. As you're running out of glycogen, right? You might use a little bit more plasma fatty acids, but right. So you're going to consume, in the end, you will actually consume less total fats at a higher intensity. If you want to consume the most fat possible, back off on the intensity a little bit, do steady state and prolong it. Do a long duration version of exercise. So some of the explanations of what this stuff is doing, you are consuming, you are taking carnitine away from beta oxidation. So if you are using glycogen and glucose, making a ton of acetyl-CoA there, you're stripping the carnitine from your beta oxidation. You are not beta oxidizing your fats. So too much carbohydrate consumption messes up the fat metabolism itself. It's not just lipolysis in the peripheral tissues can't keep up and sort of backs off a little bit. It's not just mobilization being diminished. It's also at the level of the tissue itself. This is just another picture of the same thing from a different article. Carnitine, right? So when you need that carnitine to be shuttling those fats in, you're not shuttling them in anymore. You're consuming all that carnitine over here. And so you're not contributing carnitine, the usher of fat metabolism. How do we get fat in the mitochondria so we can actually burn it? Well, the usher's gone, so nobody gets taken to their seat. None of that fat gets taken to its metabolic seat. Another thing that you could do, moving on to carbohydrate consumption as opposed to carbohydrate burning, because the higher the intensity is, remember, the more carbs you're burning. The more carbs you're burning, the less fat gets consumed. But also, ketogenic diets have been shown to increase drastically the rate of lipolysis. Now, I would not recommend a ketogenic diet for somebody who needs glycolytic metabolism, somebody in athletics who needs glycolytic metabolism. High power, high speed, you're gonna run a mile or something like that. If you're a marathon runner or you're looking to lose weight, the rate of lipolysis of how much of those triglycerides are lipolytically active, how quickly that's happening, it's way faster in a ketogenic diet. That doesn't mean you have to be carb-free all your life if you want to maximize the rate of fat metabolism, though. Let's say you cheat. Let's say you go to, it's like a family dinner and it's just nothing but carbs. You're like, well, I was trying to manage my carbs because of insulin and insulin sensitivity and to have lipolysis going all the time. But it's like, my mom's birthday and there's nothing but dinner rolls. So you eat. Something you can do. Remember, anytime you eat a bunch of carbs, your insulin releases, your pancreas, beta cells in your pancreas release insulin. Insulin is going to activate phosphodiesterase. Phosphodiesterase is going to block lipolysis. But remember AMPK, if you get AMPK to do its duty, it will clear the blood of those carbohydrates without the use of insulin. You're not going to have a big endocrine problem. So one strategy is like, I am going to be in control of my carbohydrate intake. Maybe not, ketogenic, but I'm going to manage it so I don't get insulin spikes. And then if I happen to cheat, because everybody overeats now and again, everybody sleeps poorly, everybody has a bad day, everybody's presented with a delicious looking muffin. Something happens and you eat carbs that you wish you didn't. Just go exercise for a little while after. Don't go exercise intensely for 3 minutes, because in that situation, those carbs haven't hit the bloodstream yet, and it's possible that you can get having glucose production, have the liver just dump some additional carbs into the blood, make it even worse. But sort of a prolonged exercise to activate AMPK. Remember, AMPK is going to block glycogen synthase. It's going to turn on glycolysis, and it's going to mobilize GLUT4. So it'll take care of those carbs for you, and you won't get that insulin response. At the same time, you will be getting mitochondrial biogenesis, very oxidative, improvement. So a way to manage damage of a big carbohydrate meal is just go exercise for a little while afterwards. Now, there's the debate about, should I do cardio fasted? Should I wake up in the morning and do my cardiovascular activity, get on the treadmill or go for a walk or ride the bike or whatever it is that I do? Should I do that fasted? Or does it not matter? Because whatever, the same calories I eat over the course of, let's say you eat 2,000 calories a day. When you eat the calories, does that matter at all relative to when you're exercising? Well, we know if you exercise around caloric consumption, you can mitigate some of that effect. But also, there's plenty of evidence. All of these articles are in that file folder that will be uploaded for you. The amount of fat metabolism, the contribution of fat to the total caloric consumption in your exercise. You do see fasted exercise as having a higher percentage of fat metabolism relative to carbohydrates. Now, in the long term, are we talking 6 months from now, and we've been doing fasted exercise as compared to non-fasted exercise? Not like I wake up tomorrow and what does my body look like, but 6 months from now, it depends on how much more effective this is. Especially for people who have insulin sensitivity problems, helpful. If you're like 18, 20 years old and everything's healthy and everything's normal, you have great insulin sensitivity and stuff, the difference is going to be very marginal. But certainly as people age or have insulin sensitivity problems or other endocrinological issues, this becomes much more valuable. So this is sort of population dependent, but even in resistance training, we still see a higher amount of fat metabolism. So it's not like it has to be steady state and you have to do everything exactly perfect for fat metabolism. If you're fasted, you're going to burn a few more calories of fat over the course of that workout. Sleep. So getting into sleep as a strategy. If you can't get your sleep under control, you're going to have a hard time. It's difficult to manage body fat in the presence of compromised sleep, or certainly sleep deprivation as it gets severe. So when we're looking at leptin and ghrelin, what we talked about earlier, right? Leptin, it's like, oh, I've eaten enough, my fat cells are growing, I don't need to eat right now. And ghrelin increases. Ghrelin, stomach, as your stomach is just getting like tiny and hungry, you're releasing ghrelin, and ghrelin acts in the hypothalamus and says, you go get food and just store it like you're a chipmunk, just tons of food and put it in your mouth, put it in your cabinets. Don't go shopping in the presence of ghrelin. There's a picture of me somewhere with 123 boxes of cereal. That was ghrelin shopping. That was in my bodybuilding days. So I'm like, okay, I can't eat, but man, do I have a lot of ghrelin. I'm gonna go shopping and just acquire food, and like a squirrel, I'm just gonna bury all around the house. But short sleep duration, elevated ghrelin, you're hungry all the time, reduced leptin, you're even hungrier all the time. Sleep duration and diabetes risk for lots of reasons. Impact of sleep and loss of glucose homeostasis and appetite regulation. So we're looking at cortisol, we're looking at loss of glucose regulation. There's all of these effects, and out of the way, I see cell phone activity. So there are all of these effects that sleep have on your hormones. For guys, androgens, testosterone, right? Circulating androgens. The more testosterone you have, the leaner you're going to be. And you can compromise androgens by not getting sleep. For lady folk, you're probably going to have higher growth hormone. It depends when, the response to exercise, stuff like that. But, maybe males have a higher peak at some point, but in general, the ladies in this room probably have more growth hormone right now than the guys do. And growth hormone is released at night during good sleep. Now, in the course of a day or two of bad sleep, you'll get more growth hormone. You'll rebound with a little bit more growth hormone released later. But prolonged sleep deprivation, we can start to see some problems and things like, certainly testosterone, but also growth hormone. And growth hormone is very lipolytically bolstering. It helps lipolytically active tissue. Now, when people are trying to lose weight, they'll often go on a diet of exclusion, right? They'll cut a bunch of calories. So think about if somebody has high blood pressure, what they're told is cut back salt, no sodium for you. Your BP is 160 over 88. Let's back off on that salt. But salt, that sodium, that may be their primary source of iodine, iodized salt. And so we see people on who are trying to manage risk of heart disease, especially with high blood pressure, having an iodine deficiency commonly. And you have to tell them, like, all right, go eat kelp, right? Go eat a bunch of seaweed, do something. You got to get your iodine in because you're not getting the salt anymore. See the same thing with, I'm not like, you know, this is like a vegan diet. This is just an example. There are 1,000 diets, right? I do keto. I miss a ton of things that I have to make up for. But you have to be mindful of this stuff. If you're eliminating food groups, you better keep in mind that some of those things are critical to metabolism. And so I don't care how many tricks you employ, if your thyroid isn't functioning right because you have insufficient iodine, that weight loss, they're like, oh, I was doing fasted cardio. Yeah, but like your thyroid doesn't like it, right? We need to fix that. So you need to get your sleep in order, and then you need to get the fundamentals. And so like iodine, so this is one thing that in a vegan diet, we have to be aware that iodine is commonly low. Shouldn't be, given what the sources are, right? But it is. When they do these studies, it is commonly low. Selenium also, copper is high, but like selenium also commonly low in a lot of diets of exclusion. So a few things to make sure that you have enough of in your diet. You can take it as supplements, but just to make sure that you have enough of these things so that everything else works. Because the supplements, other than, you know, basic nutritional stuff, you know, the supplements and the exercise strategies just aren't going to work if your glands aren't conducive. But for the thyroid, it's iodine and selenium. I think Brazil nuts are gross because they're the moldiest nut in the world, and I can smell a closed bat. So those will never enter my mouth. But they're the highest thing in selenium, just eat like 1. And then like a tuna, halibut, sardines, ham, shrimp. It's in a lot of things. But you can get this stuff in supplement form. For iodine, I take kelp, just like little seaweed caps, or I eat seaweed on that day. So that's my strategy: is I either eat seaweed or I take it in a pill. Most days we. Glucose regulation, insulin sensitivity. And again, don't go overboard on these. There's no evidence that having too much is helpful. Commonly, having too much is harmful. Having a deficit is going to be really hard to burn fat, though. Magnesium, zinc. Make sure you get those things in for glucose regulation. And then just oxygen carrying capacity, you know, iron. If you're anemic, it's going to be a little bit hard to have a lot of oxidative metabolism. You got to carry the oxygen, right? Oxidative metabolism, you need a lot of oxygen. So making sure the fundamentals of iron, zinc, magnesium, selenium, and iodine, and those are in place. Now, cold, what Nathaniel was asking about earlier, using the cold. Remember, the fat is innervated, and norepinephrine is one of the things that's going to cause it to be released, epinephrine, norepinephrine, using a cold exposure. So this is comparing, so here's, just look at epi and norepi. Baseline 38, that's awfully low. But epi, for the cold exposure, baseline was 38, post-exposure 110. For norepinephrine, baseline 658, the post-exposure was 1100. Now, when you look at like submaximal exercise, epinephrine goes from 180 to 311, and norepi goes from 714 to 974. So again, look at norepi with submaximal exercise compared to cold exposure. Norepi, what comes out of the nerves, and fat is innervated, goes from 658 to 1100, or 714 to 974. you're going to get a norepinephrine response and an epinephrine response, but you're going to get that response. Also, stukinate. This is another thing you will get in the presence of cold exposure is stukinate. So you remember sectional CoA. Stukinate, right next one. Stukinate triggers the activation of your brown fat in the presence of cold exposure. So burning a bunch of calories, there's a couple of identified mechanisms. One of them, stukinate, that you're going to get with cold exposure, and the other one, epi-norepi, traditional beta-adrenergic signal. So cold exposure works, and especially when you start to see pathological conditions, you can see increased adiponectin. Let's get into some insulin sensitivity, right? Decreased leptin, increased lipolysis and fat oxidation. This is what do we expect to happen in cold exposure? Improved hepatic insulin sensitivity, maybe, but glucagon secretion, decreased insulin concentration. So glucose management and lipolysis, increased lipolysis and improved glucose management with cold exposure. And you can look up the strategies, the techniques. There will be on the internet somewhere, try to find some sort of reliable source, but for the techniques, get in and out of the shower or the cold tub or whatever, just forget to turn the hot tub on in the winter, put a little chlorine or Bacquacil in it or something. Now, don't overdo the heat, right? Just like cold is beneficial. Heat, though, being in a hot environment decreases lipolysis. So think about contexts like Bikram yoga, like hot box yoga. Man, you're compromising fat metabolism by doing that. Just turn it down really cold and do yoga if you want to burn fat. All that saying, you're burning a bunch of carbs here. Now, part of it is going to be, and like, again, these articles will be available for you if you want to read through them for some more of the detail. But the oxygen dissociation curve, right? The oxygen dissociation curve is your hemoglobin carries oxygen through the blood. Metabolically active tissue is really hot. The rest of it shouldn't be that hot. Heat tells hemoglobin, in part it's heat, tells hemoglobin where to release that oxygen. If your whole body is hot because the climate is hot, you have ineffective oxygen release at the tissue. You have ineffective oxygen delivery owing to that curve, that hemoglobin oxygen dissociation curve. Now, some of this stuff we're looking at like pigs and cows and animal models, but pair-fed, this isn't a heat stress environment. This is heat stress environment here. I believe this is pigs that they used on this study. But when you look at non-esterified fatty acids, here's the heat stress, they're not releasing them. You're not getting free fatty acids released. But if you're in a normal environment, you're getting same thing right here. When you see non-esterified fatty acids, free fatty acids, this is not the heat condition, that's the heat condition. You aren't as lipolytically active. And there's a lot of reasons for this. Insulin secretion in the presence of heat, that's one of them. That's one of the proposed mechanisms, is insulin, elevated insulin. But also there's this decreased metabolic flexibility they talk about, more and more reliance on carbohydrate metabolism in the presence of heat. In the presence of heat, you become very carb hungry. And we do have data on people. This right here is people, right? Pre-exercise, this is glycogen content. So pre-exercise, post-exercise, the heat condition, they really deplete glycogen. The normal temperature condition, there isn't that much depletion of glycogen, not nearly as much in a normal environment. Another study looking at the exact same thing, but a different group of people, and it's two different conditions. It's 12 endurance trained men cycled on 2 occasions for 40 minutes. So it's just an endurance exercise, 70% of maximum O2 in an environment that's either 20 degrees Celsius, the humidity is the same in both, 20 degrees Celsius or 40 degrees Celsius. In the 20 degrees Celsius group, yeah, they consumed some glycogen, but man, they consumed a lot in the 40 degrees Celsius one. So in animals and people, whatever the species is, if you're turning up the heat, you are turning down fat metabolism, turning up carbohydrate metabolism in its place. So cold works, heat doesn't. Coffee, coffee is a caffeine, not coffee specifically, caffeine is a non-selective phosphodiesterase inhibitor. Remember, insulin promotes phosphodiesterase. Very powerfully does insulin promote phosphodiesterase. Caffeine is a weak inhibitor. You have to get a huge dose. You have to get a huge dose of caffeine to get that inhibition. But also, caffeine is an antagonist of adenosine receptors in your brain, central nervous system adenosine receptors, and that's really what most of caffeine's effect is going to be. When you look at some of these long-term studies of weight loss, caffeine does emerge as effective. Short-term, it's contextual, but the morning coffee is helpful. Okay, so we're just gonna really quickly go through a few slides that list the supplements that do and don't work. And because we're out of time, but we can talk about them more, or you can take pictures of them. I will get these lists posted also if you don't feel like taking pictures or writing everything down. But these are just the 3 articles that I used for the supplements that don't work, right? I mean, you start getting like meta-analyses of stuff. And here's the list of the ones that don't work. Now, these have effects elsewhere in the body. Beta glucans, that's great. It's an oatmeal. That's the soluble fiber in oatmeal. And it's going to bind to your bile, right? It'll bind to bile. So go eat a little bit of fat, get a little bile release. The beta glucans will bind to that. So you're gonna shit it out. Okay, bile, you're gonna have to make that with some cholesterol. Your cholesterol goes down. So I'm not saying these are terrible for everything, but for weight loss, these are things that are promoted for weight loss, not helpful. These have not emerged in the literature, test after test, to cause any weight loss. Mandy, you look like you're writing frantically. Do I leave the slide up for a second longer? Okay, okay. Now, for the ones that might work, this is a little bit more complicated in the mites, because if the state of evidence is a little wishy-washy, I have to dig into some additional literature for that. So again, all 5 of these are going to be uploaded, will be available. But here are the ones that might work. There is evidence. There is evidence. Perhaps it's not the most profound evidence that you've seen. But these ones have the possibility of helping. There is support in the literature. Yeah, that's not
What's going?
for split. I've taken that. And then for the ones that do work, here's the list of stuff that's pretty compelling in the literature for accelerating fat loss. So if you're going to take something, peppers, if we think about like icy hot, Bengay, stuff like that, it was like makes your skin irritated and stuff. I don't know if they still use capsaicin in it, but a lot of these, you can just get pure, just like your arm burn off. But you can get just capsaicin, and I've had a tube of that before. That works, but capsinoid, which is commonly marketed for weight loss, doesn't seem to work. They're both in peppers. Capsaicin is the one that seems to work. There's carnitine, we know what that is. Psyllium husk, it's a fiber. Psyllium husk is a fiber, and you can make any pastry super goopy with it. It's just really like sort of gelatinous bread. But there's evidence in literature that it works. So, all right, that's all the strategies.
Glucagon inhibit anything in import or no? Or is it just like a secondary effect because if you're releasing glucagon, you're not releasing as much?
Yeah, it's circumstantially, like in conditions in which glucagon is being released. You probably don't have a lot of anabolic inputs on board. Why is glucagon being released? does glucagon specifically have an interaction with mTOR?
There's no like competitive inhibition with the insulin receptor.
No, it wouldn't be that, but I'm not sure. I would have to do a little perusing with glucagon.